So to keep things on schedule, I'd like to go ahead and start the afternoon sessions. It's my pleasure to introduce this next session, applying precise new ways of modeling disease in animals. And just in a synopsis, the session will explore unique and non-traditional approaches to modeling disease. With that, I'd like to introduce the first speaker, who is going to be presenting remotely, and it's Dr. Nicholas Katsanis. Hopefully I pronounced that correctly. He's at Duke University School of Medicine, and he's the director for the Center for Human Disease Modeling. Welcome, Nicholas.
Thank you. Can everybody hear me okay? Yes? Good. Okay. Also, this is my apologies for not being able to be there in person. This is a unique experience. I guess soon we'll be transmitting our thoughts to each other by hologram, as we saw in the recent French election, but I will try to do my best. There's a little bit of feedback. I was asked to speak in this session, and the subject was to try to understand how we can start working with the genome, preferably the genome of a patient or of an affected individual, and try to understand what components of genetic variation might be relevant to their physiology, to their pathology. So if I take one step back, I would argue that genetics, as we have practiced it traditionally, it really is retrospective history, in that the predictive power of the genotype remains limited, some shining examples notwithstanding. However, the real question that the people that we serve, the population, the patient, expect is that we'll be able to use genomic information to make some both diagnoses, which are critically important, but a diagnosis is not sufficient when you start making some predictions and start generating some actionable items. And together with that, I am fully aware, as are many of my colleagues, I would imagine, of the sensitivities that surround information, genetic information, the interpretation of genetic information, and the misinterpretation of genetic information. And there's been a lot of work in that regard. In our unit, we have focused primarily on, at the beginning of life, some would argue that this might be the most important region, time. And the challenge in front of us, if we focus primarily on young children, can we use biological tools, functional tools, to understand driver mutations of acute and severe pathology? And when we put this structure together, the first that became very obvious to us was that we could not really do this on a single tack. So what we put together is, this is now, we're almost 5 years into the project. We really put a 4-pillar structure together. And it is my firm view that removal or elimination of these 4 pillars will undermine the stability of the entire structure. And I also should say, very importantly, that I do not consider one of the components to be more important than the other, and I view all of them to have sorry. So our biggest challenge as we went through this process was that our acquiring ability to service genetic variation accurately is poor, and by all admission, the progress we've made as a society, it's not a detriment, but to all admissions, our ability to generate data far outstrips our ability to interpret the data. So some of the questions that we always have are, is the gene relevant to the state? Is the DNA variant that functions the protein? And how can we answer this question? So as I hope some people in the audience following this conference in mind will know, some years ago we made the decision to harness advantages offered to us for this program by zebrafish, primarily with regard to transparent pathology, transparent body plans, and the ability to generate a large number of I won't go into the technical details to go too much, although I'm more questions. Suffice to say that over the years, like other technologies, we've been able to develop a number of advanced tools, and today we're more or less able to simplify between 50 and 60,000 individuals per week. And I think even though that number sounds quite impressive, it is the beginning of the journey of trying to understand the genetic variation. And the idea was very simple. Genetic zebrafish model of human pediatric genetic disorders, study the effect of genes of these systems, and then study specifically the effect of genes. I will show a couple of very brief examples. So over the years, we have generated a number of just throwing a smorgasbord here. Some of them now when I look at the already a little bit out of date again. The community has been developing tools. So for example, on the top left, you are looking at head size, and type that is encountered in a whole host of neurochemical traits, both actually syndromic. We have been able to model this with reasonable degree of accuracy. On the bottom left, you will see an example where we've been able to model the volume of the cerebellum. We've been able to be a little bit accurate, able to look at the population itself. On the top middle was a very quick example of optic nerve atrophy as a surrogate to congenital glaucoma. And in this particular case, we built a volume-based and length-based assay for the optic nerve as a direct proxy itself. I'm also showing you other examples. At the bottom right, I'm also showing you an example of peripheral neuropathy. The important thing to state here is that in each case, we have consciously restricted ourselves to human phenotypes that are organs and systems for which the direct, reasonable orthology zebrafish embryos. I fully recognize that there are other systems that could be considered orthology, and we really want to focus on structure, which is an orthology between fish and human. Otherwise, interpreting the data and translating data from one model organism is going to be very, very difficult. So the nervous system, we have actually employed some success in the nervous system, but it's not unique to that. Here's an example on old days, back in the days of 2012, of staining the craniofacial skeleton and measuring a whole bunch of surrogate features that have human craniofacial disorders, of course now with some transgenic borderline. We've been able to establish a cardiac malformations, to a subset of vascular integrity disorders into transgenic reporters. We can access some aspects of renal pathology, both the structure of the organ, but also, I'm not showing you here, in terms of like renal filtration, renal filtration rate, proteinuria, and so on. And also, we've been able to study other disorders. I like putting example of the muscular dystrophy here. The reason for this is that this particular dystrophy is a disorder that is diagnosed in teenagers and young adults. And the point to make is that not all, but on many occasions, it is possible and credible to use the developmental system to actually study adult disorders. Again, one of course has to be mindful, I think that in my mind, of the limitations that of our system, the ramp rate, but also making sure that the clinical pathology is orthologous and reasonably credible. Otherwise, the danger of a false positive or false negative must be considered. So our goal has not been just studying a few there. Our current numbers actually, we have modeled close to 800 genes. That number changes every week. But we have cleared that on the talk to the recruitment. And we're also keenly aware of the fact that it's longer to do biological modeling well than it is to recruit them and collect some of whole genome sequencing. So with that in mind, we consciously restricted ourselves to recruiting a fairly modest but well pre-selected number of families that, for whom the pipeline is useful. So at the moment, we restrict ourselves to 50 families per year. We fully understand that the need is greater than the capacity at the moment, but I think we can start on solid footing. And the results have been very encouraging. Together with many laboratories around the world, I should say that here the free flow of data, free interaction is very critical. We've been identifying on average 20 per year, together with our colleagues who've been able to expand the appreciation of the phenotypes caused by lesions in the non-gene. We've looked at oligogenic and complex inheritance. We have been able to help dissect coping of the variation. We've also had some of the drug discovery. Now, obviously, during a 20-minute presentation, I don't have time to go over this. I'm just going to highlight one breadcrumb example and something a little bit more difficult. So here's one, and it really, this is unpublished, but I take this because it's one of the more recent things done, and it really highlights how here's the family. The profile was frankly a little late, 8 months. We championed the idea that the earlier these families are pinged to the attention of the clinical and the research community, the better it is. Do a much better job at identifying those individuals, ushering in the right direction. Now, in this particular case, we had microcephaly, we had hypotonia, I read the slide. And there was a number of structural renal defects. Interestingly, there was a heterozygous NPHP1, which, when homozygous, it will actually cause nephronophthisis. But this heterozygous variant was not sufficient to cause pathology. The key that was from my colleague Bill Galantine in genetic epidemiology. So we performed whole exome sequencing on dad, mom, and the affected individual. We built up the data for rare variants, general population under 1%. We restrict ourselves to things like function. This is really par for the course. I'm not telling you anything particularly new or interesting here. Everybody does this. What we end up with is 3 sites. A compound heterozygous missensitivity in a gene called NCAP gene that has not been associated with disease before. By now, obligatory heighten. People in this game know that heighten they are perhaps the largest locus in the genome, so most humans have a heterozygous mutation. And we also found a de novo missensitivity, GLRA3, also not associated with disease. So I need to point out that, like everybody else, initially we gravitated towards the de novo missense mutation. They must have 0 to 5 mutations per exome. They're more like high delivery, other things to understand. But we mandate that every exome, and now genome, as we try to call genome, we study every gene whose segregation of alleles is with phylogeneticity. And it turned out that the de novo missense variant LRA3, based on the assays that were run, 9, and not associated with the pathology. By contrast, we started obtaining substantial evidence that it actually was a driver. The first evidence is we put NKG2 on GMatcher, and we were able to identify a second family, and the phenotypic overlap was significant. Here is the kicker. Both of these families were actually at Duke by the same investigator 4 years apart. They're completely unrelated. But this shows the volume intake in any given medical system is so high that it is not really possible to make information. So then we moved on to functionalize NCAPG2, and we were able to show that suppression results in microcephaly. You see here the image. The reason you see the image animals here is because they're being the larvae are being fed through a capillary system. We photograph automatically, and we have now also built a macro that has automatic image resolution recognition, and it automatically computes the area that we would like to capture. Very similar to calculations of your fingerprint or your retina scan. Same idea, same technology. And when we suppress the gene, we actually see a reduction of the size. We can actually rescue this phenotype by co-injecting with human wild-type mRNA. And then we can take a control allele, which is T794N. This allele is found in exact homozygosity. We would predict this allele to be benign. In the assay, it records as benign. This is important for toxicity. And then these are the three alleles found in two families. One family was homozygous for one allele, the other family was compound homozygous. So we tested all three alleles, and it turns out that all three alleles function. Expression of the allele alone, just to make sure we have some peculiar toxicity or a dominant phenotype or something like that, show them by themselves there. Of course, in DNA, we all have the capacity for CRISPR mutants, so we went ahead and we generated a CRISPR mutant for NCAP-G2, and we found that the CRISPR mutant recapitulated the morpho phenotype. I should hasten to add that because perhaps we spend so much effort in validating morpholinos, we have not observed significant discordance between our CRISPR mutants and morphos. So we have not made maybe about 50 CRISPR mutants and about 1,400 mutants. So this is, we generate more data, but I just want to give everybody a sense of what this data looks like. And we do this iteratively week in, week out. And the other point to make is that we do return these data to families that include fish data, images, and although some might think that this is a little bit overwhelming for families and patients, our experience, and we actually have objective survey data on this, is that the families are engaged and appreciative and are able to absorb this type of experimental information. Just needs to be explained well, and we need to take the time to do it. So on the second thing that I want to show today, and just try to stay close to time, is something a little different. So here is another family, and this particular kid, she was referred to as at the age of 18 months, and the index phenotype was microcephaly. When we performed whole exome sequencing, we identified again the obligatory type mutations, the novel mutations in two genes, BDG2 and NOS2. Now, again, BDG2 was predicted to be benign by every algorithm that we tested. However, functional testing showed that we did not appreciate the effect of NOS2 suppression on head circumference, but suppression of BDG2 gave rise to microcephalic embryos. Once again, I'm showing you essentially a similar experiment, suppression. I'm not going to show you the CRISPR results, but we do have, again, CRISPR mutant recapitulated orphan. So this is one expression, microcephaly. We did other experiments as well. In this experiment here, you look at the bilateral distribution of postmitotic neurons as stained by a marker for postmitotic neurons. And I hope everybody looking at this can appreciate the nice bilateral distribution in the forebrain and the midbrain, which is lost upon suppression of gene, which was restored by co-expression of human wild-type mRNA. Fails to be restored when we mutagenize valine position 1 for methionine. And the last assay we did for this one was a highly quantitative assay where that looked at the number of cells undergoing mitosis. Here I'm showing you a picture of the entire cat. The big round thing in the middle is the eye. We actually subtract all the mitosis cells there. And just using very simple image software, we will count how many cells are born. And once again, on the bottom right corner, you can appreciate that we have a number of cells born in a wild-type state. This number is reduced. When we suppress BDG2, we can fully rescue this phenotype by co-expression with human wild-type mRNA, and we fail to express when mutagenize the de novo methionine. So every assay that we've used suggests that this valence to methionine chain is benign, pathogenic. However, it is predicted to be benign. Why is that? Well, this is why. What I'm about to show you is alignment of the human BDG2 sequence with a whole bunch of vertebrates. It turns out that methionine, the de novo mutation that the child has, that we are postulating as a potent driver of severe pathology, is actually the ancestral allele, and valine is a derivative allele. So this cannot be. It cannot be that we have, it turns out, if memory serves, 56 species encoding homozygous methionine 2 and being fine, thank you, but have a child who is encoding methionine N is very, very ill. So what is going on? And this, of course, is driving the prediction alleles behind. So what is going on? A phenomenon that has been known in the biology for a little while, but perhaps we're not paying very strong attention. Again, the reason I'm bringing all this up is because as we start thinking about precision medicine, precision diagnostics, we really need to start paying very close attention to the context in which the sequence is operating. So there's a phenomenon called compensatory deviation, according to which you can have a pathogenic variant in a protein to alter its function or its structure or its three-dimensional properties or stability or ability to interact or all. That can be ameliorated by a second event elsewhere in the protein. I assure you that this has been known for a very long time, but we're not perhaps thinking about it as much as with the cornerstone. So how would you go about testing this, identifying those? Well, here's a very simple experiment. So here's the human protein sequence, and what we want to do, ask the question, we have an observation that the asparagine has been mutated to alanine in a human disease. How can this be when we have found 4 species that have this alanine? Well, the idea is very simple. We're going to look in these 4 species, and we're going to ask the question, are there any other amino acid changes that are common within the species for which the genome is permissive alanine, but are absent from the human genome? And in this particular case, we have this actually real data availing gluten. And then the test, the formal test, is that we will take the human sequence in which the alanine is acting as a pathogenic allele, and we will start reintroducing each of the valine, glutamine, and lysine, and we will seek to convert each of these things back to wild type. And that's exactly what happened. So when we look at VDG2, we take the most quantitative assay because it's also the most highly reproducible, and we can actually say, here's our controls on the left, we have suppression, and then we'll have rescue with wild type mRNA, and then failure to rescue the human mRNA encoding 141 methionine. And then on that mutant mRNA, we start adding each of the alleles that cross-species comparisons, alignment, might predict is compensatory. I can tell you we had a total of 11 candidates at site. We tested all of them, and lo and behold, turns out that the arginine 82 lysine and the leucine 128 to valine, quote unquote, leucine to valine, one of the mildest changes ever thought of. Each of these independently can fully convert a mutant human mRNA to wild type. And the most important thing here today is that just these 2 alleles alone can explain the tolerance of the methionine in 52 of the 56 species that we were able to detect, arguing that this mechanism is probably the most potent driver of tolerance of variation and of supplementation. So overall, I can give you some very quick summary data to conclude. Out of 238 families, using a combination of genetics, phenotype matching with other large centers and clinical labs around the world, and systematic functional testing, we're achieving about a diagnostic rate of 70%. In that we have alleles that we're pretty confident are causal. We have another 16% that seems likely pathogenic, possibly, but we haven't met a threshold for a variety of reasons. In some cases, there's no good variance to test the sensitivity and the specificity assay, and there's another 12% that we absolutely have no idea what is going on. And for those who have now performed whole genome studies, but I don't have the data yet. So what is the big picture? We've been running this program for 5 years. We've learned a lot going through this. But some of the things that we've learned, that our collaboration and data sharing actually, as is community engagement. And by community engagement, I mean both the physicians and the patients and the researchers and the ethicists and the ends. It has been very helpful to have strong genetics, better biochemistry, and to remain as agnostic as possible. because you never know where you're going to come from. Because at the end of the day, I just want to refresh everybody's memory that in 2001, we did celebrate one of the two papers that celebrated the completion of the first of the human genome, and it's really important that we'll continue to strive to make her here today feel better in her lifetime, not in the lifetime of your generation. And that's what makes me happy. I'll stop here. Thank you for your attention.
Thank you.